Hello everyone, welcome to the channel Microbial World. Today we are going to discuss about the structure of endospore. What are bacterial spores? Spore is a metabolically dormant structure, which means during dormancy the endospore will remain metabolically inactive and resistant to harsh environmental conditions such as heat, radiation, desiccation and chemical disinfectants. Spores are produced during unfavourable conditions such as nutrition deficiency, extreme temperature and pH, presence of antimicrobials, etc. The process of formation of spores is called sporulation. Sporulation occurs during late log phase or early stationary phase. This is because bacteria in the late log phase or early stationary phase experience various stresses such as nutrient depletion, changes in pH, accumulation of toxic byproducts. As a result, sporulation occurs during this phase. Under favorable condition, the spores will germinate and they will produce vegetative cell. The average size of a spore is 0.2 micromuum in diameter. Spores are resistant to nutrition starvation, temperature, extreme pH, antibiotics, etc. Structure of endospore. Endospore consists of 5 layers. The innermost layer is the core. Surrounding the core is the core wall or spore wall. Outside the spore wall lies the cortex. Spore coat encloses the cortex and the outermost layer is the exosporium. Now let's study about each layer in detail. Core is the innermost part of the endospore. It is also known as spore protoplast. Core has a normal cell structure as that of vegetative cell. It consists of core wall, cytoplasmic membrane, cytoplasm, nucleoid, ribosomes and other cellular materials. It contains 10 to 25% water. Therefore, the cytoplasm is in a gel-like dehydrated state. Core contains high amount of calcium and dipicolinic acid. Okay. So this dipicolinic acid in the bacterial endospore does not occur in free state. Okay. It will form a complex with calcium ions and it occurs in the form of calcium dipicolinate. And this calcium dipicolinate provides heat resistance to the endospore. The core also contains high percentage of small acid soluble proteins. Now what is the function of this small acid soluble proteins? They will bind to the DNA in the core and protect the DNA from damage caused by UV radiation and desiccation. Now how they are able to do this? It is because the binding of these small acid soluble proteins to the DNA will change the molecular structure of DNA from its normal B form to A form. Okay? And this A DNA is more compact than the B DNA and it is having higher resistance against UV radiation. And also this A DNA is comparatively more resistant to the denaturing effect of dry heat. Okay. Another function of these proteins is that they will provide nutrition and energy for spore germination. Okay. So these proteins will act as carbon and nitrogen source for the newly formed vegetative cell during endospore germination. Spore wall. This is also an innermost layer and this layer surrounds the core. Okay, so it is also called core wall and it lies outside the core cytoplasmic membrane. It contains normal peptidoglycan and it becomes the cell wall of germinating vegetative cell. Okay, so we know that under favorable conditions, the endospore will germinate to give vegetative cell, right? So during endospore germination, this spore wall will become the cell wall of the germinating vegetative cell. Okay. Cortex. Outside the spore wall lies the cortex. It is the thickest layer of the spore envelope. Cortex is very large. It occupies as much as half of the spore volume. It contains an unusual layer of peptidoglycan. Why do we say it contains an unusual layer of peptidoglycan? It is because the peptidoglycan in the cortex is less cross-linked than that of vegetative cell. Okay. The cortex peptidoglycan is composed of alanine 55%, tetrapeptide 15% and muramic lactam 30%. It is extremely sensitive to lysozyme, Autolysis plays a role in spore germination. Okay, so this cortex has a loosely arranged peptidoglycan layer, right? Thus, it can be hydrolyzed during spore germination. Spore coat. It is a thick double-layered membrane that encloses the cortex, which means there is an inner spore coat and an outer spore coat. It consists of spore-specific proteins. These are keratin-like proteins and it contains many intramolecular disulfide bonds. These proteins are rich in cysteine and hydrophobic amino acids. Due to the presence of these amino acids, the spores are resistant to adverse environmental conditions including antibacterial chemical agents. The outermost layer covering the spore coat is the exosporium. It is made up of lipoprotein and some carbohydrate. So these are the 5 layers in the endospore: core, spore wall, cortex, spore coat and exosporium. So this is all about the structure of endospore. Thank you.